need a microphone or can
We
I?
didn't put
Okay, let's start until we have the microphone. So, did you have the talk with Alex on pain already or no?
no.
Okay. So we're going to talk about somatosensation today. By the way, I haven't presented myself. I've been told that peer conditioning is one trial memory, and you all been traumatized in Engedi, so you all know my name. But nevertheless, I'm Mickey London. You have all the contact details here. Okay, is that better? Yeah, I think it's okay. So if you want to contact me, all the details are on the slides, and you get the slides on PDF, so you can take it from there, or you know where to find me because you've been in the lab yesterday. So we're going to talk about somatosensation, and it's kind of split into two talks. In the first talk, which when I went through it today again, I realized it might be too short for these 2 hours, so I'll just go at the usual pace. But if we'll finish it early, we can start with the other one, with the next one. So this is more like very basic stuff, and also the first half of the second talk is also kind of basic because I kind of assume that most of you haven't heard too much about that. But it's all covered in the textbook, so if you want to really go a little bit deeper, you can go into the textbooks, and I'm also going to cite some papers if you want to expand your knowledge about that. And at the last part of the second talk, we'll talk about more advanced stuff. So and also when we present papers later in the other session, so we kind of have half of the lectures cover the basics, and the other half is up to date. So somatosensation, in general, I can tell you that it's a little bit opposed to auditory and vision and olfactory. It's not such a very active area of research. So basically, today when you say somatosensory research, it's mostly a synonym for bowel cortex, because bowel cortex is a very active area of research, on one hand in rodents, and it's somatosensory. So people say that they study the somatosensory system, but actually they study bowel cortex, and usually they're not really interested so much in the somatosensation itself, but they're mostly interested in cortex, and just pick up the somatosensory as a bowel cortex as an area of study. But it used to be a very active research field, and there's lots known, but there's also very big gaps that I'll tell you about, and for some reason they are not. It doesn't seem that people are paying too much attention to that. So you might actually look at it as an opportunity when you're going to pick your own research subject. So I basically told you that. So what is somatosensation? Do you kind of all familiar at all with this type of sensation? What is it? Anyone? There must be someone that studied biology. Well, what is somatosensation? It's okay. So it's a sense of the body, and it's actually not one sense. It's a mixture of senses, and it's actually quite surprising that they all end up more or less in the same area of the brain. Uh, but it's a mixture of senses that has to do something with the body. Um, so if you think of vision, you can kind of, you can split the vision between rod and cone and say you have few modalities. If you're thinking about hearing, it's basically one modality that you sense. Somatosensation is actually composed of many different kind of sensations. So you feel touch, okay? You feel proprioception. Do you know what proprioception is? So proprioception is actually the sensation um, of your own body. So what is the tone of your muscles? What is the position of your joints? All these inputs that come from the body and tell you how the body is positioned in space. And not in the sense of um, balance, but in the sense of your skeletal muscle system. Okay, so proprioception, and we're not going to talk about that too much in these talks because we're going to cover it as part of the motor system in the next course, in the next part of the course, when we're talking about the motor system, because there it's very important. But it's actually part of the somatosensation system. Also covered in the somatosensation system is pain and temperature sensation. I'm not going to talk at all about pain because Alex is going to give you a special talk about that, and as well as temperature. I think he will touch upon temperature, but we're not going to cover it almost at all. So we're going to talk about touch. And you can say, well, this is just another sensation, right? Just as, we know something about hearing, we know something about vision. So, okay, so it's another sense, but it's a very special sense in many senses. And I'm going to give you some reasons. One of them is it's critical for the body integrity. I mean, that's the first way that you know if you're injured or something is going wrong with your body is through that sense. So it's very important. And obviously, that there are all kinds of disease where you have problem with feeling pain or proprioception or touch, and then people are very vulnerable and they can get hurt very easily because they don't know that they're going to get hurt themselves. It's also critical especially the part of the proprioception is critical for posture and movement. It's very difficult to move if you don't have any feedback of what your movement was, and you heavily rely on proprioception in order to coordinate that. And obviously, it's one of the most pleasurable senses. Certainly compared to hearing and vision, of course, we are very visual and auditory animals, but I don't know what passed in your mind, but pleasure is obviously part of it. So it has its own characteristics that are very special compared to others. One of them I mentioned, it's a multimodal sense. So the input that you get is not a one modality like auditory or like vision. Well, when you talk about like olfactory or taste is it's borderline. It's really difficult to define what modalities are. Here it's actually pretty easy to define the modalities, and if you rely on the receptors, and we'll talk about them, then you can see that the modalities are pretty well defined. Another very important aspect of somatosensation that it's not 100%, unique to somatosensation, but it's very prominent in somatosensation, is that it's an active sense. It's not completely active sense, but at least in part, it's an active sense. And what do we mean by that? So when you hear, some of us are talented enough to move our ears, and sometimes we move the head, right, to direct our hearing. But that's almost the most that we can do about, dealing with the sound. With vision, we can move our eyes and search for what to look for. Okay, so it is active in that sense. But in somatosensation, it's not only that you touch, when things touching you, it's when you have to move your hands, especially your hands, in order to figure out things about things that you touch. So the movement itself is part of the sensation, and this becomes extremely important in understanding this sense. So it's true that if you touch something, just touch it, you can tell something about it. But if you are allowed to move, especially with your hand, if you're allowed to move your hand, you can tell, you can extract much more information, and this is actually being used. It's also true, and I'll show you in a second, not only in humans, in other animals as well. And it actually means that in some ways we have to think about this sense in a different way than we think about other senses. Okay? So active sense, and this also hints to the fact that, again, we think of ourselves as visual animals and auditory animals, but the fact that you have these signs in museums, don't touch, is telling you something very deep about that sense, that it's extremely important to us. So if we go to a museum and you're only allowed to watch things, it doesn't give you the same experience as if you're allowed to touch it. And it's kind of it's a tendency of us, of everything we do, we want to touch it. It's also becoming very important recently because we all have these devices, which are called touch devices, right? Even though there's nothing touching about them, right? It's more like tapping devices rather than touch. And in a sense, I don't know if you had the chance to use the old type of mobile phones before this became available, but in all devices you could ring someone just having or text someone having the phone in your pocket. You can't do it today, and the reason is because it's not a touch device. You can't tell what you're doing just by doing touch, right? You have to look at it in order to know what it's doing, and it's actually an active it's an active research area in engineering. How do you make these screens such that they will also be able to allow you to feel texture on them? Okay, so there are all kinds of different materials that you can actually play, with electronics and make these things having different textures, and this will change a lot the way we will. I think also the way you actually feel about it, emotionally. The fact that you can touch something and it reacts to your touch, it makes a difference. Okay, and lastly, and this is something that I previously included in the talk, but here I just left it as a link to a YouTube movie or a TED talk by a guy called Olaf Blanke. That he has a very good talk that you can see online, and he also has there's a guy in Hadassah that was in Gedi, Shachar Arzi, that, if I'm not mistaken, did his postdoc with him. Somatosensation is a sense that in some ways tells you what is your body. I mean, what is you? What is yourself? So is that hand that moves here. This is an object that does something in the space. How do I know it's me? How do I know it's not just a floating hand that moves here in space? What makes us feel that this is us? It's a very deep question, and eventually, it's something to do. I mean, we don't know the answer for that, but it seems to be something to do with the fact that we control it and it reacts in the way that we expect it to act. Okay, and it's something that we learn. But there are all kinds of abnormalities around that thing. For example, there are these out-of-body experience. So out-of-body experience is something very strange, right? When you are in the middle of an operation and suddenly you see yourself outside your body and look at yourself. That's not what we talk about, but you can do tactile stimuli that goes on. There are all kinds of illusions that you can do where you get like a sequence of tapping along your hand, and then it continues along the other hand. If you position your hand in a certain way, you will feel a tap in the air between your hands that is kind of, you feel it, a tap. It's not there, right? It's an illusion, but you feel it. So you can feel things that happen out of your body in a way, just because you create an illusion. So it's related to that, and this is kind of a little bit comes, it's beyond what we normally call physiology or neurophysiology, but it's certainly part of neuroscience. And you're welcome to watch the movie. I was trying to get an icon for each one of them. This is a self-portrait of Lucian Freud with איך אומרים נכד? נכד של פרויד. Yeah. Okay. So if you look at the classical view of somatosensation, very similar to many other senses, there are 4 stages that we kind of we separate it into 4 stages. The first is transduction, is actually making the physical quantity that is related to the sense, like light in vision. So here it will be something like mechanical force, for example, okay, or vibration. We make it into a neural substrate in a way. Okay, we let it into the neural system, and from there on it becomes a neural thing, and then it has to be transmitted somewhere, because we don't feel with our fingers, right? We feel somewhere else. It's integrated, so at the beginning the input is transmitted, and then it's being integrated, and up to this point we can kind of more or less describe it in reasonable way. This point is, as you have seen already, I guess, in many other senses, is a big gap. It's not completely understood, because perception is borderline between consciousness and awareness and all kinds of things like that. So these are somehow, we separate it here to integration, which is something that we can explain in terms of connectivity and activity, and perception, which is more related to the psychological phenomena. So we will talk about that at the other part a little bit, but not too much. And one of the reasons I think that somatosensation is unique as a field of study is because as opposed to many other senses that we have like direct access. When you think about vision, you have more or less direct access to what you see. With somatosensation is a little bit more, undefined. So when you, for example, study the whisker systems of a mice, you know a lot about the anatomy, about the physiology, but it's very difficult for you to imagine how the mouse is using its whiskers. You don't have any introspection about that. The only thing you can think of is like touch with your fingers, but it's obviously not the same thing, touching with the fingers and feeling whiskers, right? And this is an advantage and disadvantage. The disadvantage is, of course, that you don't have this introspection, so it's really difficult for you to come up with an hypothesis. The advantage is, on the other hand, that you don't know and you don't have this introspection, so you have to be a little bit more objective, right? You have to kind of come up with an hypothesis that you can, that it doesn't have to do with what you feel about things, and we know that sometimes these introspections can lead us to really wrong conclusions. Okay, so this was the classical view, and the modern view is more complicated because it takes into account, it's basically complicated without gaining much, but I would say, but it realizes that this is not a feed-forward loop. I think you talked a lot about it probably with Shaul. Shaul was giving you vision, right? So he was talking about reverse hierarchies and all this kind of stuff. Right? So in somatosensation, this is certainly, in a way, it's even more natural. And it's enough to think about what I told you, that if you want to, if you know if this is rough or smooth, you would do that, right? So it means that you actively look for something. Okay, you give a command to your motor system to test an hypothesis, right? By moving your finger over something like that. So obviously there are all kinds of loops and feedbacks that are related. So it's not information coming this way. It's actually a lot of the information here is actually sought by the system. The system is actively seeking the information. And this is an illustration, but another illustration, and I put it in small diagram, but I will show you a large version at the next talk. This is the loops that we know of in the barrel, not in the barrel, in the whisker sensing system of rodents. Okay, so there are loops already there are loops already at the level of brainstem. That the nuclei that control the movement of the whiskers. So whiskers are moved by the mouse, are controlled or getting feedback already from the nuclei that receives input from the whisker. And this closing one loop, and there are also loops that going through the cortex to the brainstem, and there are all kinds of intermediate loops in the thalamus, in other areas, and you can see that this is crazy system. So even if you knew what each one of these nuclei and stations are doing, it seems almost impossible to understand how the whole thing would work as a system, because you have all kinds of feedbacks with all kinds of delays and all kinds of interactions, and information can go, it can go this way, but it can go also this way, and it can go this way, and it's almost impossible to untangle. So this is something very important to understand about the fact that you have feedback in the system. Okay, so after we got like an overview, let's go into the details. So as I guess you haven't talked about spinal cords at all, right? And who knows anything about spinal cord? Okay, some of you don't, some don't. Okay, so if you think about vision, hearing, taste, and smell, they're all in the head, right? The somatosensation comes from the whole body. So a lot of the information is going through the spinal cord. The spinal cord, you have, I guess you haven't done any anatomy as well, right? So the spinal cord itself is part of the central nervous system. Okay, so all the neurons in the spinal cord are part of the central nervous system. They're basically part of the brain. Okay, it's like an old type of brain, but it's part of the brain. Um, and the information to the brain or to the cortex and to the other areas of the brain is going through the spinal cord, and some of it's directly through the brainstem to the brain. So we'll talk a little bit about it. We'll talk in much more details about the spinal cord in the other half, because obviously spinal cord is also very important for movement and motor control. So we'll talk about it more there. But nevertheless, I will mention few of the things that are critical to understand. So let me first tell you the simple stuff, and then we'll kind of talk about spinal cord in more details later. So the spinal cord has, we split it into the dorsal part and the ventral part. The dorsal part is what kind of on the side of the back, and the ventral part is what towards our front. And in general, the spinal cord is, and it's also split into two areas. You have the gray area. You can see it here. Better, no, I don't know. Yeah, here, you know, whatever between this butterfly. You see this butterfly? Okay, so this butterfly is a gray area. And as in the brain, gray area means the place where the cell bodies are, and all the rest, what's around it, is a white area, and this is where the axons are. Okay, so the spinal cord is divided into the gray area and the white area, but inside of them there are still subdivisions. So the major subdivisions that interest us is this part, which is called the dorsal horn. That the dorsal part is this side, right? So this is called the dorsal horn and the ventral horn. So the ventral horn is where the motor neurons are, and these are the neurons that control the muscles. And the dorsal horn is actually, it's mostly composed of interneurons. So it's neurons that are limited inside the spinal cord. But it's also the place where axons from the body, mainly from the skin and from the muscles, are getting into the spinal cord. And this is where the input comes into the somatosensory input comes into the spinal cord. Okay, so in brief, the input comes, if you look at it here, say this is a finger. You have some touch receptors in the skin. So this is like a very general overview. Okay, the input comes through an axon to the spinal cord. Now, what does that mean? It means that at some point there's a neuron that, differently to what you usually know about neurons, that they have dendrites and axons, these neurons has only axons. And this axon is going to both ways. So it goes all the way to the finger and also into the spinal cord. So it's basically the same axon. And it's not that the cell body emits a spike and then the spike would go this way and then that way. The spike is actually initiated at the end point, at the finger. So when you touch something, there is a spike generated where you touch it. This spike goes back in the axon, so it's for him it's actually for the spike, for it is forward, but it goes in the axon. The cell body is not important in that sense at all. It's just you know, for biological reasons. It's an axon. It needs to it needs to synthesize proteins. It needs to do all kinds of stuff. So this is what the cell body is for. But the cell body is not taking part in the information processing here. The spike goes directly through the axon, continues into the spinal cord, and then in the spinal cord it does two things. It can synapse on interneuron. It can make a synapse on an interneuron, but at the same time it will go up inside the white matter of the spinal cord. Okay, so it will send one branch into the dorsal horn, but it will also send a branch up in the white matter until it will get to some place, and this place depends on the type of axon that we're talking about. And for example, this could be in the medulla, which is here, right? So it can go all the way. It can go here, right? It can go. It could be an axon that starts at my toe, going all the way to the spinal cord, come in here, go into the spinal cord, and then go all the way to here. It's a very long axon. Okay? And there it synapses on a cell, and this cell will send that axon and cross to the other side, because you have this laterality that touch on the right side of the body goes to the left part of the brain. So here it crosses the midline to the other side, and then continues to go up in all kinds of other stations until it goes to the thalamus. Okay, and in the thalamus it will synapse on a neuron, and from that neuron there will be an axon that goes to the cortex. And cortex is a whole different story. We'll talk about cortex later. Okay, but so if you think about it in terms of vision, for example, so in a way, you have like the retina is from here, we're going all the way to the medulla. Okay, it's a 1 meter long thing. That's the retina of that thing. Okay, because it's the same photoreceptor that you have on your finger for that sense. It's a sense receptor, but it's the same one that goes all the way almost to the brainstem. Okay. And then it synapse, and the thalamus is like LGN. So this thalamus, it's a different nucleus in the thalamus, which is called VPL, ventro-posterior lateral. And then it goes to the cortex. And the cortex is somatosensation 1, just like primary visual cortex. So this will be primary somatosensory area. So that's overview of how the information is getting through the system. Okay. So again, so I haven't mentioned one thing here, and this is these cell bodies that I told you about. You see that this is an axon that starts in the skin, goes back, a very long axon, and it's not that it goes into the cell body and continues. It just continues. Okay. And the cell bodies themselves are sitting in a place called the dorsal root ganglion. So this is a ganglion. A ganglion is just a bunch of neurons, okay, that are sitting outside the spinal cord, but inside the vertebra. Okay. And the axons of these neurons are getting into the spinal cord, and then they split into the into the dorsal horn, and up into the spinal cord in the white matter. Okay, now if you look at what happens here is that these neurons send the axon into the ventral horn. The motor neurons are sending into the dorsal horn. The motor neurons are sending the axon from the ventral horn outside, and once they meet after the dorsal root ganglion, they bunch together and goes together to the organ. So this already tells you that you have input from the skin and output to the muscle of the same organ will go together. So these are some mapping there that goes from the same axon. So it's not the same axon, but from the same area, the input is going at the same track as the output. Okay, so this is a mapping then. Um, that's what I wanted to tell you about that here.
So from the ventral horn, there is only output to the muscle, or there is also input from the
No,
system?
the input all comes through the dorsal root ganglion. The output all goes through here. So, for example, I told you that some of the input is coming from the skin, but some of the input would go from the muscle because we'll talk about it next year, but there's what's called muscle spindle, which are sensors of the stretch of the muscle, and they are also going through this nerve, but they would not go this way, they would go this way. Okay, they also have cell bodies in the dorsal root ganglions. What it also tells you is that the, the, the spinal cord is, in a way, if you look at it, it's one very long thing, okay? But the ganglions are separated, so there's a ganglion for each vertebrae. So it means that this segmentation, this imposes segmentation on the spinal cord. So there are segments. Okay, we'll talk about it in a second. Okay, so what kind of receptors do we have that actually start the whole thing here? Okay. So there are receptors for touch. We'll talk about them soon. Okay, and there are all kinds of different sensors, not that many, something like 4 that we'll talk about. There are sensors for thermal sensation, for hot and for cold. There are sensors for pain, and this is Alex will talk with you about them. So I'm not going to touch them at all. And there are sensors for proprioception. Proprioception, which are there are few kinds. There are ones which rely inside the muscle, which are called muscle spindles. There are some on the tendons, which are called Golgi tendon organs, and there are some in the joints, which are feeling, the position of the joint, and also some friction. Um, but all of that, we'll talk about that next year. So for today, we're concentrating on that. We're not talking about that almost at all. I can tell you that we don't talk about that also because we don't know much about it. It's kind of strange, but some of these receptors are not even known. No one knows exactly how that works. Some of it are, but some of it are not. Okay, so we talked about that, right? There's something happening in the skin. There's some sensor here that causes an action potential to start. This action potential, this is going through some, we'll talk about that in a sense. We call it mechanoreceptor in touch, because this is what creates the spike. Okay, this one goes as a spike through the spinal cord, and from there it goes up. So how do we actually transduce the touch into a spike? So the truth of the matter that we don't really know that for sure. Okay, we have some hypotheses, and we are starting to understand that. But it is known. One thing that is known is that there are some ion channels that, because of mechanical stretch, it's a mechanical thing, they will open and let ion in. And if you have high sodium outside, low sodium inside, you stretch, you open the receptor, sodium comes in, you get a depolarization. It's an axon. So if you have depolarization in the axon, it will spike, and the spike will just propagate on the axon. Okay. So one option is that you have just you have ion channel sitting in the membrane. You have a stretch of the membrane. This imposes force on the protein, and the protein opens. Okay, so that's one option. This is one. Okay. The other option is that the protein itself is connected to something that connected to the skin, and then when the skin moves, it will open the channel. Okay, will physically make, the protein will be stretched and will create a conformational change that will open this channel. Another option is that you have something that is sensitive to stretch, that will use some second messenger system inside the cell. So this will active some protein, that will active some other protein, that eventually will contact the protein of interest and opening. Okay.
Something I thought about now. So why aren't all the ion channels mechanically opened when I mean, because if I stretch something, then even if they're supposed to be ligand gated, I guess they should be pulled apart.
Not necessarily. It depends how they sit in the
No.
how they sit in the membrane, right? If they are bound to the cytoskeleton or not. Like if so, if, for example, a channel is bound in two places to the cytoskeleton and then stretch, it will open the channel. But if it's just floating, around, not bound to anything, then you stretch the biolipid layer and nothing happens to the channel. But as I said, I can't answer that too well, because I don't think, I certainly don't know, and I don't think, as far as I understand, it is largely unknown. Okay, because it has to do with um, it has to do with things that we only now starting to be able to do. You need to kind of know the crystal structure of the protein. And not only the crystal structure of the protein, you need to know how the protein is actually sits in the membrane, what it is actually bound to. And these things are just getting there now. I mean, people have hypotheses about many of these things, but they haven't done most of them. I'm going to show you in a second, but there was a question before.
I just don't understand why it is so hard to clarify which is. I mean, it sounds like a very clear question, and it doesn't sound hard to verify.
Um,
Maybe I don't know, but
so here is an example. Okay, again, I told you this is a largely unknown subject, and I think, I don't know if this CU will present this paper, but that's a paper that we presented last year in the student presentations. Okay, and this is a paper from 2010, and it's based, and it's, if I'm not mistaken, it's published in Science, which means that it's high profile publication because it's a new finding. Okay, and it's two proteins that were kind of found to be mechanically activated. So basically, it's not a very long paper, but it's basically they cloned these proteins in some cells, and they show that when they apply force on the cell, they can measure current through these channels. Okay, and they call it piezo 1 and piezo 2. Anyone knows why? So do you know what's piezoelectric? So I think I've shown some of you in the lab. There are certain type of ceramic materials that if you apply voltage to them, they change the shape. They can move, for example. Okay, and they can do it very, very accurately. So all the guys that are working in nano, for example, they need to move things very, very precisely. They're using that. It also works the other way around. If you apply force to them, they will create a voltage. So, for example, the early types of acoustic guitar amplifiers would put a piezo on the guitar, and then when there was vibration of the string, it will actually translate it into a voltage. So it's a different way to. I mean, in music you can do it also with microphones, but piezo is something that kind of translate mechanical force into voltage or into current. Okay, so this is why they called it piezo 1 and piezo 2, but Again, they were only cloned in 2010, which means that people are working on that, but apparently it's hard. It's not so obvious. I mean, how do you know which of the 2,000 proteins that you have in the nerve ending of your finger uh, are the mechanoreceptor? It, it, it just needs work, right? Um,
There are three mechanisms that could be, one of them could be possible, or
so I think this is more like what we can think of, what we can imagine, but maybe there are even others that we don't know of. But it sounds like these are the obvious candidates, right? Okay, so this is one thing. Um, there are other type of receptors that I told you about, like these pain receptors, the thermal receptors. Um, the thermal receptors, for example, seem to be activated by. So there are actually channels which are called TRP channels. TRP channels are famous for also for pain, also for other things. And if I'm not mistaken, the first person to clone these channels, and certainly a very big advocate of them, is Baruch Minke from Hadassah. I don't know, he's part of ELSEC. He's one of the senior people here in ELSEC, and he found it first, I think, in the Drosophila, in the visual system of the Drosophila. But it's a family of receptors. Some of them are sensitive to, for example, chili. In terms of capsaicin, Alex will tell you a lot about that. He has a very interesting story about that. And some of them are, for example, TRP M8 is activated by MINT, but also activated by cold temperature. So you can, and this is why when you eat mint or use a toothpaste that uses some ligands that are bound to these channels, that's why you feel cold. Okay, so it activates the same receptor, which means that it signals through the same axons, and it's impossible to tell whether it was mint or cold, right? Okay.
okay. So what is a TRP receptor?
It's an ion channel which is working. It's a sorry. I think it's not an ion. It's an ion channel, but works through a second messenger. So I think it has like a ligand binding thing, and then there's a second messenger system that. Just not, I forgot actually. Um, it's only a channel that I'm sure about, but it also has a very strong second messenger system bound to it. So now, let me answer later. I don't want to say something stupid. Okay, do we have time to? Yeah, do you want a break or do you want to go straight? What, break? Okay, so let's take a break.
יש לך מלא תאים של מי יש שם?
you've been here as well. Thought you've been in this class. No, no. Okay. Um, so let's go on. So first of all, TRP TRP channels are just ion channels uh, that are let's say cations in, so sodium and calcium. And these are the channels that they're called transient potential. So TRP is for transient potential because they are creating depolarization when they're activated. In the Drosophila, these are the ones that responding. So there is a complicated mechanism that I don't remember completely, but the activation by photons activate the TRP channel, and this is what creates the depolarization due to the activity. And what I'm not completely sure about, but we can ask Baruch, is that there is a complicated mechanism that involves second messenger that lets you be very sensitive to a single photon. So the whole idea is that you kind of you can sense a single photon because there is some amplification mechanism that one activation of the channel will translate into a cascade of events. Okay, so back to touch. We're going to talk now a little bit about the encoding and the sensors itself, the sensors that are responding to touch, and we're going to touch upon all these aspects, spatial aspect, temporal aspects, the intensity, and some other more complicated things that are related to the sense of touch. So we have 4 types of receptors in the skin that are responsible for touch, and we split them into 2 type of categories. One is the superficial ones and the deep ones. So the superficial ones are these 2, Meissner corpuscle and the Merkel discs. We'll talk about it in a second. And the 2 deep ones are Pacinian corpuscles and Ruffini endings. So this is one type of separation between superficial and deep. And there's also a different separation which is rapidly adaptive and slow adaptive. So the Meissner corpuscle and the Pacinian corpuscle are the rapidly adaptive, and the Merkel discs and the Ruffini endings are the slowly adaptive. And this relates to the temporal aspect of the sensation. Okay, so there is a very clear structure here, and you have these ridges in your skin. You know what used to use your fingerprint. Okay, and the Meissner corpuscles are sitting along the ridge, and the Merkel discs are sitting below. You know, this you can see it here. I don't know how to call it, but underneath the main ridge. The Pacinian corpuscles and the Ruffini endings are sitting very deep in the skin, and there's another difference, which is the Meissner corpuscles. So all the superficial ones, they don't have one axon per receptor. So if you have one receptor here, you have another receptor here. They are all kind of coming up from the same axon, okay? Which will become one axon. You know, they connect and then they continue as one axon. The Pacinian corpuscles and Ruffini ending, you have one receptor per axon. The axon doesn't split when it comes to the skin. So this means that if you activate specifically this Meissner corpuscle or this Meissner corpuscle, there's no way that your system will know that it comes from a different place. But if you activate the Pacinian corpuscle, you know that it's coming from this specific one. On the other hand, you obviously notice that you have many more of those than those, so it's all kind of balanced up together eventually. And this is more kind of modern way to look at them. You can express in the medial corpuscle GFP, or in the Merkel disk you can express RFP, and you can see how these are actually organized and why they're called disks inside the inside the skin. That's another way to look at it, where you see a scanning electron microscope of the skin, and this is after histology, and you see these ridges here, and the medial corpuscles are stained and they're kind of ordered along the ridge of your skin. Okay, so this is just to show that. And this dictates something about the receptive fields of each one of these receptors. So in general, all your sense of touch is coming up from these 4 different receptors. Okay, and the receptors have receptive fields. So I don't have to explain to you now what receptive field is, right? You know that enough, right? So receptive field in this case would be the area, so like the spatial receptive field, we have spatial and temporal. So the spatial receptive field will be all the areas on the skin for which a single nerve that goes to the spinal cord will react to. Okay, so we go and touch different areas, and all the area, and this nerve, this axon that goes to the spinal cord will react only to touch in that area, in that purple area or this red area, and you see that the receptive fields of the superficial layer receptors are quite localized and small. On the other hand, the deep one, the Pacinian corpuscle and the Ruffini ending, have pretty big receptive fields, so they sense from a large area of the skin. And there are much, there are much smaller number of these receptors compared to these ones. Okay. Now, and you also kind of, if you magnify one of these receptive fields, you see that there are hotspots in this receptive field, and this is because at every point where you have a receptor, you have a hotspot, right? And then, and I told you already that these receptors are kind of connected together to one axon, so it's not completely uniform in terms of, like, if you really want to very, very accurately map the sensation, it's not completely uniform. Okay, so this is one aspect of that. But there's also important, that except from the fact that we'll talk about in a second, that there are separation into slowly adaptive and fast adaptive, you have to also remember that so what makes a receptive field is not only the space, it's a combination of we're going to talk about it soon. It's a combination of space and also of the properties of the receptor. So the receptor is also, which receptor it is and what it is reacting to is also important in terms of understanding this receptive field. And the other thing to notice is that when you touch something, it's not only the contact point that is important for touch, because the contact point is actually creating a deformation of the skin, depending on how much force you apply, and this could actually activate receptors, stretch receptors, or mechanoreceptors that are not in direct contact with the object. So here, the skin is stretched, so receptors here will also respond. So it's not only it's not always the receptors that are in touch with something that will respond to a texture. So a better, people have tried to study that. And as a side note, I will tell you that nerves could be, people can decode from nerves in humans. Okay, you can put electrodes in the skin of humans and put the electrode inside the nerve. When I was a student in Dan's lab, there was a woman from researcher, and she was actually studying motor system, and she would record from the nerves of humans, and she didn't have too many subjects, so she would usually record from her own nerves. And she was very good in, and she was she was here for 3 months and was convincing us all the time that it's not painful, and she knows how to do that. But we politely refused. But it's good. It's completely possible to put a very thin electrode in your skin, and sorry, not in the skin, in the nerve, and record the activity. And one way to do, I mean, one way that people have done that, and I think the link for this paper is here. You can download it. It's a review paper. What they've done is that they made this device where you put your finger on, and there's some membrane over here, and there's a pin that can come out, and it can go in certain direction, and they control the direction of the pin and also the force, and they press your hand down on this thing, and they kind of can measure the position of your finger, okay, and also the force that is applied, and at the same time they can record from your nerve and map the responses, and they can also kind of play with, for example, whether there is friction. So if this is only latex, or if you add some powder which makes it a little bit more smooth and less friction, how would that affect the mechanics of the skin due to this force? We can go into all these details. I'm not going to talk about them too much. I just want you to appreciate that it's a complicated transformation. Okay, it's not just like lighting a pixel in a certain photoreceptor. It has all kinds of effects on the surrounding. One thing that you can do is actually measure the sensitivity in terms of the minimal distance between 2 points that you can actually distinguish. Okay? And this is the highest sensitivity is in the fingertips in humans. It's around 2 to 5 millimeters, which actually sounds completely strange. You know, when you think about it, you naturally feel that you have a much better resolution in your finger. Okay, and this is the reason. It's actually true that you have a much better resolution, and the reason is that active touch. Okay, so you can, if you move things around, you can actually distinguish really tiny things. Also because you integrate from many places, it's not only one. But if you just do the blind test of distance between location, it's around a millimeter. I think in young people it's a little bit more like 2 millimeters in older people. And there are areas in your back, for example, that are very insensitive to this test. Now, just as a side story, when I prepared to this talk, and I went through the presentation, I looked at this thing and I suddenly asked myself, what did they actually take it for? And with a little bit of search, I found out this guy. His name is Weinstein, and he has this paper. It's not a paper; it's like a publication. It's more like writing up. How would you say? It's a text of a talk that he gave for a celebration of 50 years of his research. And I actually, from this point on, for the presentation, and I hope it will go well, because I stopped because I went and read this paper, and I haven't finished. So I really urge you to read it because it's interesting. It's very interesting. So I'll just tell you 2 anecdotes. One of them is that he tells about. So apparently he was doing this, and he invented a device that is actually being used consulting people with Hansen disease that I never heard of before, at least not under this name. And if you look that up, you find out that this is disease in which there is it's called leprosy, and it's a disease where there's a problem in the skin, and slowly people are losing. I mean, they're losing the
tips of the
sorry?
the tips.
Yeah, they lose the sensation in the sensation of touch, and this leads to actually very bad consequences, because they also, due to injury, start losing parts of the fingers and things like that, because they don't notice that they have a problem. And it turned out that the device that he was using became a very useful tool for these clinicians to evaluate how sensitive, what's the sensitivity of the patient to touch. So he actually tells the story that he was a soldier in World War II, and he was actually hurt very badly. He had burns all over his body, and he was kind of asking himself why he doesn't feel pain after these injuries, and this actually led him to study somatosensation. And then he has another interesting, I think very interesting story. A bit stupid but interesting. So he was an undergrad, and he got a honor fellowship, and as a fellow he had to do some research as an undergrad. So he went to the department, psychology department, and he was working with some professor, and he was basically studying sensation of pain to heat. And he had this device, and he was very enthusiastic, and he would kind of pick up any student on the campus and would measure the threshold for sensation. And one day his wife came, and they went to dinner, and he told her, just before dinner, let me just get you as a subject as well. And she didn't want. She said, it's going to be painful. So he said, no, I'm going to put it on the lowest level. It's not going to be painful. Don't worry. Everybody is doing that. It's not a problem. And he just started the experiment, and she jumped, and she was she was shouting, and he was completely surprised because it was on the lower threshold. So she told him, he asked her what's going on. She said, it was very painful. He said, how come? Everybody else was doing it. No one felt anything. You are really far from threshold. She said, okay, why don't you try it yourself? And he was trying it, and it was very painful. At the end of the experiment, of course, I went to dinner. He was very occupied. We went through all the data and tried to figure out why these two guys are much more sensitive than everybody else. And then eventually, completely randomly, decided to sort the data out by, obviously, before computers or anything, just by the time of the day of the experiment. And he found out that there's a perfect correlation between the time of the experiment and the threshold. And then he could not figure out, how is that like, our sense of touch is changing through the day.
But which was the most sensitive?
What?
When were you most sensitive?
At evening, of course, because they went to dinner. Just before
Oh,
dinner, they were the most sensitive. And the conclusion was, the first thing you should actually test is your experimental device. So he went and made like an experiment where he actually left the device on through the day and measures the temperature of the device. And basically, what happened is that the device was flawed. And if you would leave it like for just a little bit too much, just the only way the device was kind of calibrated is right after you make the measurement, you turn it on. So you cannot actually use this device more than like for a second, because then it comes out of calibration. And then then it goes on and on. Of course, it had to. This was a device that was used in many universities and was invented by a very prestigious professor, and they basically just by finding out that thing, he got appreciated enough to go onto an academic track and doing 50 years of research. But it's very personal. It's written very nicely, so I urge you all to read it. I will put it on Moodle if you want. Interesting stuff. Okay, so this was spatial resolution, now temporal resolution. So you remember that I told you that there temporal aspects, and we are splitting these receptors into slowly and rapidly adaptive receptors, which means a slowly adaptive receptor is something that when you give a stimulus which is long, it spikes through all the time that there is a touch, and it increases the frequency as fast as with the amplitude of the stimulus. Very natural. And of course, at the beginning, it has high frequency and it adapts, but it adapts slowly and it spikes throughout the stimulus. And the rapidly adaptive ones will act as a change. It's more like a derivative of the stimulus. So they will act where you have a change in the stimulus rather than when you have a constant stimulus. So that's the difference between rapidly adaptive and slowly adaptive. I guess it's not the first time you see something like that. Right? And we'll talk about it a little bit. Have you heard about Adrian? Adrian. Okay. So we'll talk about it next, in the next semester when we talk about proprioception. But Lord Adrian was considered in a way the father of neural coding, if you want. He was the first person to quantify these things when he was kind of recording from stretch receptors. And this is a classical picture of something that, frequency coded for amplitude. Right? You see a low amplitude stimulus, high amplitude stimulus, and the frequency encodes for the stimulus. Okay, so this is a classical picture that Adrian got first, but this specific one comes from another very famous guy who's called Vernon Mountcastle. Anyone knows him? Wasn't even mentioned by chance. So Vernon Mountcastle is a famous guy studying somatic sensation, and he's a person who came originally with the idea of the cortical colon, or actually with the evidence for the existence of cortical colon, which in some sense Hubel and Wiesel got the Nobel Prize for, at least for some of it. He was, in some sense, some people believe he should have been included there. But anyway, he was the first one to classify them in that sense. And this comes into, you can, if you do this experiment where you put like braille texture on a drum and you rotate the drum and you touch it with one finger and you record from axons, from slowly adaptive or rapidly adaptive axons that are connected to this axon, you would record, so each dot here is a spike. Okay, and here you see the different responses of the different type of receptors. So the slowly adaptive ones will act, see, like as long as the point is touching your finger, you have spikes. Okay, the rapidly adaptive ones are kind of more like reacting to the friction. So it goes like you have vibration due to the fact that you have friction. So they spike and stop, spike and stop, spike and stop. And slowly, the ruthenium ones that are very, very deep, almost don't respond at all. And the Pacinian corpuscle, which are very fast, they respond all the time, but because they have like this large spatial receptive field, you cannot actually tell if you had 1 point or 2 points. They just react more or less the same. Okay, so in this case, if you want to really decode the system, you have to rely on this Merkel system, okay, which are the slowly adaptive, superficial receptors.
How is this data exactly taken?
I'm not sure if it's been taken from a human or from a monkey, but I would guess it's a monkey, and you will put an electrode in the nerve, okay, and you can kind
Inside
of
one axon or on the...
you put it in the nerve, and then you do spike sorting, because you can kind of go on the finger and kind of stimulate very lightly, so you know you are in superficial layer, or very strongly, so you know you're in deep layers, and then you can, I'll show you in a second. Okay, here you can see that you can separate the, you can separate, by recording from a nerve, you can separate what type of receptor you're activating, whether because they have like different properties. So if you put like, if you put a stimulus which is weak, okay, but then you get constant response from that, it would probably be a slowly adaptive superficial receptor. Okay, if it reacts to fast, but it's light touch, it will be, and also on the side of the receptive field, you can distinguish that it comes from this one. So you can actually know which axon you're recording from. And then you can kind of compose these things. Fine or not? Well, you can't actually kind of record from one axon in the hand. You wouldn't find it, I guess. Okay, so you have to go where the nerve goes. You stick an electrode in the nerve, and then you start searching for the axon that responds to it. Okay, so these receptors are sensitive to. You can kind of characterize them by by the sensitivity to frequency. So this is one of the things that Mount Kessel was doing, and what you do is you apply a device that is doing a vibration at a certain frequency, a mechanical vibration. Okay, and then the rapidly adaptive receptor, the Pacinian corpuscle, they are responding phase log to the stimulus. They don't always, like in a high frequency such as 200 hertz. So here, for example, you see that this is like the threshold, right? This is the threshold. So this receptor here this is a logarithmic scale, and this means that the lowest amplitude of vibration that you have to apply, they will respond, to 200 hertz. So this is like auditory neurons response scale. Okay, so you have here the frequency, and here you have the amplitude of the sound. Now here, instead of sound, you get here like a mechanical vibration. Okay, so the Pacinian corpuscle has this type of receptive. How do you call it? Tuning curve. Okay, this type of tuning curve, which means that they are more sensitive at high frequencies. Okay, and the others, like the Meissner corpuscle, are sensitive in lower frequencies, but they are all much more sensitive than the Merkel ones, which are slowly adaptive, and they are really responding only in a very low frequency. Okay.
Do you the
now,
one?
sorry?
Do you the
I think it's completely out of the scale. I think they are like about 5 hertz, so they would be somewhere here. Anything above that you don't even measure. So they respond only to very, very low frequency. These are the base. These are the base sensations when you stand in front of a big base thing.
Right, when they say frequency, what do they mean? Could you like tap fingers, tap?
So I would think something that vibrates. So it vibrates at 200 hertz or something. It's like if you touch a speaker, for example.
Right, but that's not very natural, is it? Because
What?
no, the
200
frequency.
hertz.
Right, but usually we touch things that are stationary, like a
Right.
floor or a
Right.
wall.
It's true. I don't actually. I mean, you can see it here, right? That you get. I think it's more. It's more things like that in terms of friction when you move. So if you want to feel some texture, okay, and you move your hand, it will create this, these frequencies because of that. So it's true. I don't exactly know what these ones are coding for in natural life. On the other hand, they prove to be extremely important because you can see that if you have response at 200 hertz for tactile vibration, it could only come from the Brazilian cortex. So it's a very good way to separate a specific modality of somatosensory sensation. And this being useful, bound by Mount Russell, when we'll talk about it when we'll get to cortex.
One more question. The dashed line, is it a behavioral threshold?
Yes, exactly. So this is quality of behavioral threshold, and it's kind of, they show that it's kind of composed by the different ones, and basically in this case it's Meitner and Brazilian corpuscles that makes all the one.
So it looks like you have access to the complete information.
Yes.
Okay.
But I don't know what is. It's a good question. I don't know what it's used for. Okay. So the next thing is transmission. So once the sensor are sending these axons, where do they go? So did you talk about peripheral axons at all? You probably didn't. Right? I already told you that all these axons are bound into a nerve. So if you would cut the nerve, you would see that it's actually kind of, it has subdivisions, okay? They have all these complicated names that you would not remember, so it doesn't really matter. But it's like there are bunches of nerves, of axons that are bound together. And each one of them, you see, this is like an enlargement of an axon, a myelinated axon in this case. And there are blood vessels between them to supply them with blood. And they are bound in some membranes, and then these ones, like each one of these bundles, would be bounded together with other bundles, and this would make the nerve. This is what it would look like in a real cut. Okay, and you see all these axons, and they have different diameters, and we'll talk about it in a second. And again, it's a different illustration, but each one of these axons has myelin and internodes. That you already learned, right? Okay. So it has gaps between the nodes, which are pretty long. It could be very long. And the conduction, at least in the myelinated nerve, is saltatory conduction. Okay, so it's kind of it jumps from node of 1V to node of 1V, as I think that was clear before. Okay? No? So do you know why do we have myelin in node of 1V? So how come you don't know how the property, the actual potential property? You do?
We do know, yes. But he done this.
He done this. Okay. It's a very impressive structure if you think about it. I mean, this is the axon, and it's really wrapped with sometimes 200 wraps of myelin sheet. It's very impressive. And this picture I like particularly because first time I went to neuroscience, and I found it very interesting because it was one of the first time people stained with antibodies specific ion channels in the axon. And what you can see is that the node of Ranvier knows this high density of sodium channels. But it turns out that it's not just high density of sodium channels in the node. There is segregation within the node, which is only 1 micron, between the sodium channel and the potassium channel. So the sodium channels are in this stripe, in this green stripe, and the potassium channels are on the side in this parallel node. This is where the potassium channels are, and also, this is a myelin, the red is the myelin, and the potassium channels are in the parallel node, and also a little bit in what's called the juxtaparallel node, so in these areas under the myelin, but when the myelin, so the myelin kind of start wrapping before the node, like slowly, so this is called the juxtaparallel node. And it turns out that there's a complete molecular machinery that holds each one of the channels in place. And just as a side note, from cable theory point of view, there is no point in moving like potassium potassium channels 1 micron away from the sodium ions. It's one compartment. There's no way these two things should be separated. But it does turn out that in some conditions this could be important. I'm not going to tell you which because it's not part of this class. But it could be interesting at least in two directions. One, when you think of like what happened to the signal near the soma when the spike starts, this could be important. And the other thing is that there are today some theories that sodium channels are not only voltage dependent, but they're also talking to each other. So when one sodium channel is open, it actually increases the probability of very close by sodium channels, other sodium channels to be open as well. And this would make a model of, the Hodgkin-Huxley model would be invalid in that sense, okay? Because now the channel is not only sensitive to the voltage, it's also sensitive to what other channels around it are doing. So before it was also sensitive to that, but only through the voltage. But here they are structurally cooperating, if you will. Okay? If anyone is interested in that, I can tell you some stuff.
You actually identify something that connects the channel, because
it's not coming from from molecular and structural. The evidence are not structural and molecular. They are more physiological, and they are very weak.
But it sounds as if you have a channel that's a calcium channel, then it makes sense that if one is open, then neighbors will also be open just because there's calcium in that area, not because they're related to that channel.
Right. But here we're talking about sodium channels. Okay? And they're not sensitive to the sodium. They're opening to sodium, but they're sensitive to the voltage. So normally you would say that they're sensitive to the voltage and they all feel the same voltage, so they all have the same probability to open. But you can actually build a model where the probability of one channel to open, depending on what its neighboring are doing, insensitive of the voltage. For the same voltage, the probability is getting higher the more other channels are open. And there are evidence for something like that for other types of channels, not for sodium channels. And for sodium channels, just from the kinetics of how spike starts, it seems that it better fits a model like that than the usual Hodgkin-Huxley model. Okay, so now there are different types of fiber. I already hinted and told you that there are different sizes of diameter. Okay, so one classical way to separate them is if you apply some stimulus, say you put an electrode in the nerve and you give an electrical stimulus. If it's a small stimulus, you would get a recording will feel something, a weak sense. If you ask a person what does it feel, it will tell you no. It will be very noticeable. If you increase the amplitude, you'll get this first wave, but not. It will be obviously bigger because you give a stronger stimulus, and after that there will be another wave. Okay, and this is basically this wave is carried by the biggest axons, which are called A alpha, and the A alpha are the biggest myelinated axons of the somatosensory system. Okay, you have also